Okay, so we're going to move on and finish this subsection. And one thing that was brought to my notice by a student in the class, there's a typo here. Pyruvate kinase is the enzyme that catalyzes the reaction. This reaction, ATP, is a non-competitive inhibitor of PFK. I mean, ATP is a non-competitive inhibitor of PFK, but in this case, this should be PK to keep it correct. So just a quick correction. All right, so pressing on. Fermentation. So we talked about this right here in a previous video. The pool of NAD plus NADH is constant. Therefore, if the NADH increases, the NAD plus decreases. In glycolysis, one reaction transforms a NAD plus to a NADH. So let's go ahead. And the reaction is written down here, but let's go ahead and take a look at this. What are we talking about? All right, let me go ahead and erase some of this stuff. All right, let me erase this. Okay, so the reaction is right here. So as glycolysis proceeds, and remembering that NAD plus and NADH are tied to each other, meaning that if NAD plus goes down, or let me put it in a different way, as NADH concentrations go up, NAD plus concentrations go down. Now, if there is no oxygen, oxidative phosphorylation will not happen. And once oxidative phosphorylation is not happening, what will happen is that the citric acid cycle will stop working too, so pyruvate will not be transformed to acetyl-CoA. Okay, so that's sort of a chain of events that will happen under anaerobic conditions when there's very little O2 or no O2 available. So glycolysis is the only thing left to make 2 ATP from each glucose molecule. Now, this is not very efficient. It's very little ATP for breaking down a glucose at least to some level, but it is the only thing left for the cell to do. So the problem is, as glucose goes through and glycolysis is happening, well, this one reaction right here is also churning, right? This reaction that we were pointing to right here, right? This reaction is constantly taking NAD plus and reducing it to NADH, to the point that if this was the only thing available to the cell, glycolysis, you will run out of NAD plus. And if you run out of NAD plus, then this reaction cannot occur because NAD plus is a reactant in that reaction. And if this reaction stops, then the two reactions right here and right here that actually produce ATP from ADP will not occur, and thus the cell doesn't have any ATP. It starts to run out of ATP. So glycolysis by itself will not work under anaerobic conditions. You need something else. So let's go back. In aerobic conditions, NADH will be transformed back to NAD plus in oxidative phosphorylation in the mitochondria, which will reconstitute the NAD plus pool. So under aerobic conditions, when oxidative phosphorylation is active, the NADH will be oxidized back to NAD plus, so you start to reconstitute the pool of NAD plus needed for glycolysis. Okay? Without NAD plus, glycolysis cannot continue. So what do we have? Under anaerobic conditions, the NADH is oxidized to NAD plus with the following reactions. So we have two reactions here. This reaction which produces lactate or lactic acid, this first one, A, this is the one we see in most animals, or all animals. And the second one is seen in yeast, which is the making of ethanol and CO2. So let's look at the first one. What is happening here? So you can see here that NAD plus is reduced to NADH. So under anaerobic conditions, the second reaction is activated. And I'm just going to put a circle around this second reaction so you guys can see it. Here, the NADH is oxidized back to NAD+, so that it can be reused by glycolysis. What is the reaction? The product of glycolysis, pyruvate right here, is transformed to lactate. And this is a reduction. You can see that right here, this double bonded oxygen is turned into an OH group. And that reduction is then reciprocated by NADH being oxidized. So in this way, you get your NAD+ back, so glycolysis can keep occurring under anaerobic conditions. Now in the second reaction, the one that occurs in yeast, you have the same thing. You have a reaction right here that will reconstitute the NAD+ pool. In this case, it's two steps. Pyruvate initially loses a carbon and becomes acetaldehyde right here. And then acetaldehyde is reduced. You can see the reduction. You have a double bonded oxygen right here. And this is an OH group now. So that's the reduction step. So acetaldehyde is reduced to ethanol. And that reduction is reciprocated by the oxidation of NADH to NAD+. And the NAD+ pool is reconstituted in that way, and you get ethanol. And of course, CO2. So the important thing to note is fermentation is glycolysis plus A or B, and A or B being one of these reactions. So when we say what pathway or metabolic pathway does the cell use under anaerobic conditions to produce ATP, it isn't glycolysis. That is an incorrect answer. Why is that an incorrect answer? Because if it was just glycolysis, the cell would run out of NAD+, and no more ATP would be made. And if there's no more ATP, the cell can't maintain its structures and etc., and it would fall apart and die. So it is glycolysis plus one of these reactions, and that is called fermentation. So the correct answer would be either fermentation or specifically saying glycolysis plus the transformation of pyruvate to lactic acid or pyruvate to ethanol and the reconstitution of the NAD+ pool. Or you can just not say all of that and just say fermentation, right? So I would just say fermentation. There's no reason to say that whole thing. Okay, so that's fermentation. So fatty acid oxidation, structure of triglycerides. I've drawn out the structure of triglyceride right here, a triglyceride molecule. And triglycerides are hydrophobic. Even though there's a carbon double bonded to an oxygen, this is still a very hydrophobic molecule. Now, in cells, triglycerides are kept in these organelles called fat droplets. In certain cells, there's more fat droplets than others. For example, fat cells have a lot of fat droplets in them because that's their primary job of storing triglycerides. Now, triglycerides are a really good source of energy. In animals, triglycerides and fat cells are used for energy production in other cells. So the triglycerides are hydrolyzed. So what is this hydrolysis reaction? Basically, this hydrolysis reaction is plus 3H2O, giving me 3 fatty acids plus the glycerol. That's the hydrolysis reaction. Of course, there's an enzyme involved in this. There's always an enzyme involved. So in fat cells, the stored triglycerides are hydrolyzed, and then the fatty acids, right here, enter the bloodstream, and then they go to cells that actually need them. And once they get into that cell, they enter the cellular respiration and are used to produce ATP. Now, the thing about fatty acids is that they enter cellular respiration at the citric acid cycle. They skip glycolysis. So thus, they can only be used under aerobic conditions, because under anaerobic conditions, the citric acid cycle and oxidative phosphorylation are shut off. So fatty acids cannot be used because of that. And what they do is fatty acids are immediately turned to acetyl-CoA. So let's take a look. I have a structure of a fatty acid right up here, and let's look at how fatty acids are turned to acetyl-CoA. Okay, so the fatty acid has a CoA group attached to it, so it becomes an activated fatty acid, and it enters this cycle. Now, this is not the citric acid cycle. Just to be super clear, it is not the citric acid cycle. What this cycle is doing, it is basically removing 2 carbons at a time to produce acetyl-CoA from the fatty acid. Now, if you look, this right here, if I cleave that off, that is an acetyl-CoA. Okay, so all this little cycle is doing here is cleaving 2 carbons off at a time and then adding the CoA group to the end. So let's take a look at what's happening. So we have a reduction right here. FAD to FADH2. That means an oxidation step has occurred. The oxidation occurred right here where a double bond was formed. Now, here you have an H2O coming in and you have an OH group added to that activated fatty acid. Now you have another reduction step, and NAD plus is turned to NADH. And what happened to the molecule? Well, now you have a carbonyl group instead of an OH group, and that's the oxidation step. Now what you have is this part right here, I'm going to circle it with green, is going to be cleaved off, and then another CoA group comes in, and you get back basically to where you started from. Except the fatty acid, the activated fatty acid, the fatty acetyl-CoA, is shortened by 2 carbons. So basically, if I have a fatty acid that is 14 carbons long, then I can get 7 acetyl-CoAs from it. And then, of course, the acetyl-CoA enters the citric acid cycle, and all that NADH will later be used by oxidative phosphorylation, as we will see in section 2. Glycolysis and the citric acid cycle are central metabolic pathways in the cell. Intermediate products of glycolysis and citric acid cycle are building blocks used by other pathways to reconstitute other molecules such as amino acids, nucleic acids, monosaccharides, lipids, and more. So all this picture is showing you is that we have glycolysis here, the citric acid cycle here, and all those molecules that are made from breaking down glucose can be used to make other molecules. They can be building blocks for making other molecules. So smaller molecules to making bigger molecules, those are going to be anabolic pathways, right? This whole thing is a catabolic pathway, and we talked about how catabolic pathways are linked to anabolic pathways, and this is just showing you how that is possible, or showing you an example. Other metabolic pathways can feed their intermediates into glycolysis and the citric acid cycle. For example, amino acids and etc. can enter glycolysis at different points and be used as an energy source. It doesn't need to be just glucose. Because of numbers 1 and 2, glycolysis and the citric acid cycle are central catabolic pathways that directly or indirectly feed into many other metabolic pathways. And this picture right here is showing you the complexity of all the metabolic pathways and the fact that they are connected to each other in some way or the other. So you can see that glycolysis right here and the citric acid cycle, which are also highlighted in red, they are in one way or another attached to all of the other metabolic pathways that are within the cell. And vice versa, the other metabolic pathways can feed into glycolysis and the citric acid cycle. Alright guys, I will talk to you later. Bye-bye.